to our final panel as we continue our celebration of 15 years of the Radcliffe Institute, 135 years of Radcliffe, by probing another area of intellectual fervent, this time in the biological sciences. Our final panel of the day, What is Life? The Science and Ethics of Creating New Life in the Laboratory, will consider the promise and perils of scientific research that occurs at the intersection of biology and engineering, a new area of investigation made possible by scientific advances in gene research. Our moderator today is Professor Eric Lander, President and Founding Director of the Broad Institute of Harvard and MIT, Professor of Biology at MIT, and Professor of Systems Biology at the Harvard Medical School. Eric is co-chair of President Obama's Council of Advisors on Science and Technology, in recognition of the extremely important work he and his colleagues have done on the Human Genome Project. You can read more about Eric's many accomplishments on the program card on your seat. Now I will turn the reins over to Eric, who will introduce the panelists and frame our conversation this afternoon. We are enormously grateful to Eric that he has agreed to moderate this afternoon's panel, generously bringing his wide and deep personal experience with the science and ethics of new life in the laboratory to the Radcliffe Institute.
Well, welcome to everybody. It's been quite a day, and it's really exciting to close with this very forward-looking topic. We have an extraordinary panel here today. We've got 4 faculty from Harvard and 2 faculty from MIT amongst the 5 people, because I'm actually both, and so that's how the overcounting works there. And I will introduce them as they come up, but it's an amazing group. We have a chemical engineer, a chemical biologist. We have a systems biologist, a genome biologist, that's me, and we have a bioethicist and law professor. And it's also a very lively group, as I think you will see. So the title of this panel is, What is Life? The Science and Ethics of Creating New Life in the Lab. Now, we didn't write the title, but that first 3 words, what is life, is actually iconic in our field. The Nobel laureate physicist, Schrödinger, wrote in 1944 a popular book called What is Life? And it was an amazingly important book because, now, by the way, let's make clear, it is not, the panel is not about what is the meaning of life, right? That was, at best, a previous panel on literature. We do not intend to address that here because we lack qualifications. We're not going to address the origins of life, many other aspects of life. If that is not your, if what is life is not your destination, this would be a good time to exit the plane. But we're going to address what is life. And Schrödinger, in that book, basically said, the question is, how can the events that take place in a living organism be accounted for by physics and chemistry? Or do we need some other sets of laws of nature other than physics and chemistry to explain life? And of course, the thing that life does that is most remarkable is it faithfully reproduces itself. One generation gives rise to the next, and, you know, as they say, plant a radish, get a radish, not a Brussels sprout. And so, why is that? This was the famous philosophical text, I quote there. Why is it? What is it about life that allows it to transmit information this way? And that's what Schrödinger was asking in 1944. And he hypothesized that it must be some interesting bit of stable chemistry that had some repeating structure, but not so much repeating structure that couldn't be new. He called it an aperiodic crystal. And of course, within a decade, that aperiodic crystal was clearly DNA and the DNA double helix. He nailed it in the abstract and interested generations of scientists from other fields to come into biology. So I'm going to give you the brief tour from 1940 to the present. It'll go real fast, so hang on. In the 1940s, scientists really did show that the substance of heredity was DNA. That if you transferred DNA between microorganisms, it could carry with it hereditary information and change the properties of the bacteria to which you transferred the DNA. That wasn't true if you transferred proteins or transferred other things. The DNA contained the hereditary material. How could it possibly do that? Well, in the 1950s, we got an answer to that from Watson and Crick with the double helical structure of DNA. 2 strands of DNA, each a complete template for the other, containing information in a linear 4-letter code. We, of course, in our world take for granted the idea that you can code anything you want in 1s and 0s or 3-letter codes or 4-letter codes. In the 1950s, people weren't so used to the idea that any information at all could be written in a simple code. But Crick and Watson showed us that this 4-letter code was all there was. That was what DNA was about. And this magical secret of replication was merely untangling the 2 strands of a DNA double helix and using each as a template to reconstruct a partner strand. Now, in the 1960s, the code by which that DNA was translated into the proteins of our body, a very simple 3-letter lookup table was worked out called the genetic code. And you got to the 1960s and people could say, if I knew the DNA sequence, I could figure out the protein sequence. And it was a triumph and Nobel Prizes were awarded. But the problem was you couldn't read the DNA sequence. So it was a little bit of a Pyrrhic victory. But in the 1970s, people began to work out ways to clone and propagate individual pieces of DNA, the so-called Recombinant DNA Revolution, to take DNA from any organism and stitch it together with DNA from another organism that would allow it to propagate, and use those pieces of DNA to produce enough molecules to read out DNA sequence. Now, the 1970s was pitifully little bits of DNA sequence, but the idea became in everyone's mind by the 1980s, maybe we could read out the complete DNA sequence of organisms. The idea of a human genome project, it was kind of nuts. It would cost $3 billion, people calculated, but by the end of the decade, they decided, let's do it. Let's spend the $3 billion and sequence the whole genome of the human. I and many others got involved in that project, and by 2000, we were able to report out, the year 2000, we were able to report out essentially the complete sequence of the human being. It did end up costing about $3 billion, but what's remarkable is in the 14 years since then, the cost of DNA sequencing has fallen from $3 billion to $3,000, a factor of a 1,000,000-fold decrease, which has never been seen in any technology ever before. And by next year, it'll be down from $3,000 to about $1,000. That is stunning. The amount of information that's being read out right now is amazing. It allows us to begin to understand the basis of disease. Genes for breast cancer, BRCA1 and BRCA2, where mutations give rise to a high risk for breast cancer. Genes for diabetes, for schizophrenia, for Alzheimer's disease, for colon cancer. It allows us to begin to ask, how can we use that information to treat patients? Early mammography for breast cancer. And if someone has the mutations for breast cancer, mastectomy actually produces a 90% decrease in risk of death. It's a fateful choice, but it has a huge consequence there. Colon cancer, early screening. All sorts of treatment decisions are increasingly being based on DNA, and we'll see more and more of that. You can do more fateful things than that. You can use this to select life. If you know you're carrying a genetic disease in your family, you could choose to have pre-implantation genetic diagnostics and decide which embryo to implant, and decide not to have a kid who is certainly going to die of spinal muscular atrophy at 9 months, or not to have a kid who would have Huntington's disease, or maybe not to have a kid who wouldn't face late onset Alzheimer's disease. Which choices are appropriate choices to make? But then beyond simply selecting life, where we are right now and what this panel will focus on is the ability to, and I use this in quotes, improve life. To say, hey, we could do better. We could take a bacteria and teach it things it couldn't do before. We could teach it to make insulin to give to human patients. New life in the laboratory. We could teach rice to make vitamin A and provide vitamin A to vitamin A deficient people in the third world. We could teach bacteria to make oil and perhaps make fuel for people on farms. We could evolve entirely new functions that have never been seen before through evolution in the laboratory. I'm describing all sorts of things this panel does actually in their work. We could begin to treat the rules of life like microprocessors. The ability in the 1970s to write general rules for making microprocessors gave rise to this incredible creativity of computers in the 1980s and 1990s and young kids writing code for computers. Will this next decade see us do the same thing, writing code for life? New organisms with new capabilities, and if you think this is impossible, let me just tell you in the last year and a half, tricks have been discovered in bacteria that let you go in and edit human DNA with precision. What should we be doing? Oh, and in addition to reading and understanding and treating and selecting and, quote, improving, there's owning. Who owns any of this? This should be enough of a framework for a panel. It's up to them to answer those questions, but you have now come all the way from what is life to the questions we face today, and as I hope you will see, every one of these things has urgent, important things that we have to do for people's health, for prosperity, and at the same time has fateful, deep ethical questions. Unfortunately, there are no clean, easy answers about we don't want this technology because we could do something wrong with it, because all the things we care about for health and for food and for energy, we need it for. So how to make these decisions? I will start, I believe, with Linda. Linda Griffith, my colleague at MIT, who is a professor of biological and mechanical engineering. And I will not read her whole impressive bio that is here. But Linda, what do you say?
First, I say it's wonderful to be back here at Radcliffe, where I spent a wonderful fellowship year resolving a dilemma in my life. On the one hand, I strive to create life in the lab through tissue engineering. On the other hand, I strive to stop the creation of life. And it's here at Radcliffe that those two things really came to resolution. I want to share that with you. So when people think of tissue engineering, they often think of regenerative medicine. And if you're old enough, you might remember an experiment I got involved with when I was an assistant professor, creating a human ear on the back of a mouse. This has gotten into an icon in popular culture, but it really was emblematic of trying to regrow tissues and organs in vivo. And I turned my attention to trying to regrow livers, really challenging, life-threatening problems. These kinds of challenges required integration of biology and engineering, and I became involved in starting a new department at MIT, biological engineering, doing engineering based in modern molecular life sciences that spreads out into electronics and all kinds of things, and is now MIT's 4th largest undergraduate major at MIT. But what I want to focus on, I still work in areas of regenerative medicine and liver and bone tissue engineering, but I want to focus on some branches that came off around the time in the mid-1990s when we were just starting this department. And one of them was in the area of liver tissue engineering. I had an aha moment. I'm spending my life trying to regrow tissues for transplant, regrow organs, but is that really a good goal? What's the barrier to really solving liver disease so people don't need transplants? Animal models can do so much, cell culture models can do so much. What's lacking were more complex models of human disease, models that incorporated immune systems, 3D, blood flow, and so on. A great engineering problem. So could we build models that would help us understand and prevent liver disease and get rid of the need for transplants? And that's what I turned my attention to and still work in this area. It turns out it's a great engineering problem, not only for the building the model, but also for the systems biology side. You got to frame the question of what's causing the disease and figure out how to get information out of your model. Okay, so this led to a whole, that field has taken off in the past 5 years. There's a DARPA program. I'm part of it now. I run a $35 million 5-year program to build interconnected tissues and organs on a chip, and it's really exciting, and we continue to work on that. But how did that actually get me to Radcliffe? I'll come back briefly to that topic later. So about 7 or 8 years ago, I was at MIT, and I got asked by some really great friends of mine who are on the corporation to participate in a Women in Science event and to talk about my research. And I usually don't like to go to Women in Science. There was a lot of things. I was really busy, but these were really good friends, so I accepted. And then they asked if I would talk about how my research particularly impacted women. I found this question actually kind of annoying because I worked on liver and bone. Everybody has these. And, I didn't view myself as a woman engineer who's working on women's problems. I just like one of the guys working on stuff. But these were great friends, and it really made me start thinking about the ways that, as an engineering faculty member at MIT, maybe I could do things, tangible things, to help women. I was a little put off by a lot of political correctness things at MIT at the time, I admit, that were making my life unpleasant. So the question was annoying, but it did make me think. And it particularly was annoying for another reason, and I did have an unresolved issue. Even though I drive motorcycles and consider myself a tomboy, something set me apart, and that is I had infertility, and I'd been through in vitro fertilization. And that process really, really changed my life. At the time I went into it, I was working with George Daly, an expert in embryonic stem cells. Where do you get the cells to create these organs and culture to do drug development and model diseases? You needed stem cells. So I was working with George, and I thought, aha, I'll go have IVF, I'll get extra embryos, we'll make our own stem cell lines, because it was back in the day when you couldn't do that. So this is just great. Then you get in there and you look through the microscope and you see your own embryos, and you cannot imagine what a force of nature the maternal instinct is. You can't reckon with it, it's not rational, and it completely changed my outlook on embryonic stem cells. These were my kids. I worried about them being in the incubator when I was home in my warm bed, and I hope the CO2 was properly being controlled and all that. And so it really gave me a different sense. And this, more than anything else in my professional life, made me awkward sometimes around men, colleagues like Rudy Yanish and Harvey Lodes, who I love dearly. I teach with them, but they had not been through the experience the same way I did and couldn't understand that I respect people who want to donate their embryos and make ES cells, but I just couldn't do it. So it made me think back to this issue of, are we putting our effort in the right place to grow a liver or understand liver disease? Why are so many women creating these embryos? Most infertility is female infertility, and 5 million IVF babies, more than 5 million have been born to date, and many times that number of women never could get pregnant. And so it made me wonder, what could we do from an engineering sense maybe to help these women? Could we stop this production of embryos, which I think is can we stop the suffering of women who go through this? And so the leading cause of infertility in women is a disease called endometriosis, which is characterized by endometrial cells growing in the abdominal cavity, sometimes invading. And it's a lot like cancer, but not exactly cancer. It's extremely debilitating. It often has symptoms and onset in teenage years. Poorly under-recognized. 10% of women have this. So how many people are in here? Let's just say 300 women in this room. 30 of you probably have endometriosis, whether you know it or not. And it's characterized in the textbooks as being a disease of women in their 20s and 30s. And so there's a delay to diagnosis of average of 10 years in the US. And in fact, around the time I was being asked this question, my own niece who was a teenager in Atlanta, was missing school because she was so sick. Her OBGYN told my niece was making it up because she wanted to get out of going to school. She was a National Merit Scholar, fabulous, 1 in every 5 years student. She was really sick. She was diagnosed with stage 3 endometriosis finally when she was 16. So this led me to Radcliffe. I decided that NIH only spends $8 million a year on studying endometriosis. Gazillions more dollars are spent on treating infertility. So I decided to apply for a Radcliffe Fellowship and decided to start a center at MIT to focus on women's gynecology problems, which are all icky. Men at MIT have no problem talking about breast cancer, but anything that might involve tampons or menstruation, no. And so, it kind of hit me, could we build an engineering center, bring a scientific engineering language where guys would pile on and say, hey, this is really cool, this is and it is a great problem. Let's frame it in engineering terms. And so we got a foundation grant. I had my year at Radcliffe. Unfortunately, got diagnosed with very aggressive breast cancer in the middle of it, which then, it was unfortunate because I missed a lot of my Radcliffe time, but fortunate because it made me think about stratification of disease. I got stratified instantly, unfortunately, to a horrible category, but molecular markers determined my prognosis and treatment. Nothing like that existed in endometriosis. So I took that experience, and we brought the field of systems biology. I got about $1 million foundation grant, and we started bringing systems biology in and analyzing immune networks in the inflammation. We managed over the past 3 or 4 years to figure out how to stratify patients into categories, in fact, using some of the math that came out of the Broad Institute, non-negative matrix factorization, to look at immune networks. In fact, just published a paper earlier this year that's the first molecular stratification of endometriosis patients. It was really well accepted at the recent conference in Brazil. So So we've been successful in this goal, and I think what I've tried to do is give you a flavor for these kinds of conflicting things. We hope to eliminate the need for creating ES cells. Thank God iPS cells, induced pluripotent stem cells, have come along to resolve that dilemma otherwise. But thank you, Radcliffe, for hosting me to help sort out not only the scientific but social issues and thinking how to go forward in these two areas.
So now I turn to David Liu with the "Can you top that?"
Well, you can't come close.
David Liu is a good friend, otherwise I wouldn't have said that. That's
Thanks, Eric.
No. David is a professor of chemistry and chemical biology at Harvard. He is a senior associate member of the Broad Institute and a good friend. And in his spare time, he runs evolution in his laboratory. And you always wanted to know who is in charge of evolution, so I can introduce David Liu.
Yeah. All right, thank you. I think I lack some of the necessary physiology to even try to top Linda, so I won't try. And in fact, just to set expectations properly in contrast with the title of this session, I will disclose at the start that no one in my lab is currently trying to create life. But all of the projects in my lab manipulate fundamental features of living systems to try to study and illuminate and advance biology and medicine. Okay, so in the next 6 minutes and 24 seconds, I hope to show you how the study and manipulation of these fundamental features of living systems, the most fundamental features that arguably define the difference between living and non-living, can offer benefits to society even separate from whether or not we are in a much more sensationalistic topic, creating life in the laboratory. Okay? Okay, so over the past 15 years at Harvard, our lab has become obsessed with, in awe of, inspired by perhaps the most fundamental and powerful of features defining living systems, which, as Eric already spoiled, is the ability of life to evolve. And we became interested in developing ways by which scientists could evolve new drug-like molecules instead of using living systems to evolve new proteins or new biological molecules. So we developed, over the past 15 years, a method by which we can harness some of the most powerful features that underlie biological evolution to discover not new biological molecules, but rather new drug-like molecules. Okay? And the key is figuring out a way to translate DNA sequences not into RNA or into proteins, what Eric referred to in his introduction, but rather to translate DNA sequences into synthetic molecules, into drug-like molecules. So we scratched our head for 4 or 5 years trying to figure out how to do this. And in the end, a key solution, not the only solution, but a key solution was very simple, that we can rely on Watson-Crick-based pairing to bring together chemical reactants to produce these drug-like molecules. And in the end, what you've achieved is you've taken a sequence of DNA and used it to program the synthesis of a drug-like molecule. Okay, so what have we done with this method? 30 million people in this country have diabetes, probably several dozen people in this room have diabetes, statistically. And virtually all patients with diabetes take drugs that are either insulin, increase your sensitivity to insulin, help you make more insulin. But a strategy that has never before been even tested in an animal is the strategy of inhibiting your body's natural tendency to degrade insulin. There is an enzyme in your body, it's uncreatively named insulin-degrading enzyme, that degrades insulin. Okay? And scientists for 60 years have tried to figure out a way to inhibit this enzyme with the idea that if you can keep insulin levels higher, diabetics will have better control of their blood glucose levels. And those efforts have failed for a variety of complicated reasons. But we used, one graduate student in my lab used this platform and within a few months had evolved, in essence, a molecule that can inhibit very potently and selectively the degradation of insulin in an animal. He then used that molecule to validate for the first time that slowing down the degradation of insulin in a diabetic animal, a diabetic mouse, can actually help alleviate that mouse's diabetes. And so in the process of harnessing some of these fundamental features of living systems, all in a test tube and then eventually in cell culture and eventually in mice, we validated a new target for the treatment of diabetes and have focused people's attention on a series of molecules that, if developed further, could serve as leads for future diabetes drugs. As a second example, one of the fastest life cycles, most worthy of our awe and admiration, I think, as nerds, is that of a simple virus. There's a virus that infects bacterial cells that can transmit its genes, make progeny, evolve, all within a span of 10 minutes. Okay? So it could have gone through several generations in the time between when Eric started talking and now. Our lab has co-opted, has repurposed this viral life cycle to dramatically speed up the evolution of scientist molecules, molecules of interest to scientists in the laboratory. So we can now evolve proteins that have biological or medicinal relevance using this system, and we can do so at a rate that's about 100 times faster than was previously possible. So we've used this system, which is called PACE, to evolve proteins of therapeutic relevance, to study biological questions about evolution, about drug resistance, for many proteins, about 10 different classes of proteins now. And then finally, I'll just summarize work that I'm constantly in awe of. It's only a field that we've recently begun to perform research in, but it's a field that Eric referred to, which is that of genome engineering, something that many of you may have heard of. It's now more commonly called genome editing for reasons that I don't understand. But basically, the story begins with bacteria that defend themselves from infection by cleaving foreign DNA. Okay, so the question you should ask yourself is, bacteria don't seem that smart. How can they determine what's foreign DNA versus what's their own DNA? And the answer is, every time they get infected with a virus or with a foreign piece of DNA, they install a piece of that DNA in their own genomes that makes an RNA that guides a relentless machine that cuts up DNA. Okay? That machine only cuts DNA that matches the sequence of the RNA, the guide RNA that is made as a record for past infection. Okay, so scientists over just the past few years have co-opted this machinery to now cleave DNA sequences that we program the machine to cleave rather than that the bacteria cleave to fend off viral infection. What is the utility of this? This capability makes practical, for the first time, I think, in human history, the possibility that we could address genetic diseases at the level of our software, at the level of our genomes. Virtually all methods to address human disease either address the symptoms, or maybe the leaves of the tree or the bark of the tree. Very, very few therapies actually address diseases that are based on genetic changes to our genome at the level of our roots. And so that possibility of engineering our own genomes to improve human health, to address perhaps many of the 20,000 or so human genetic diseases, is just an incredible, mind-blowing possibility to me. So I hope I've convinced you, or at least stimulated some thinking about how we can co-opt some of the most fundamental features that drive living systems, that drive their evolution, and use them to offer society and science some real benefits. Thank you.
So next we're turning to Pam Silver, and she really does create life. I mean, she's in the business of creating new organisms that can do new things. She is a professor of systems biology at the Harvard Medical School. She's a founding core member of the Wyss Institute for Biologically Inspired Engineering, which I think is an inspired name for an institute. And what are you creating in the lab, Pam?
Thanks, Eric. First of all, I'd like to celebrate Radcliffe and thank Radcliffe also for the year I got to spend here and to ponder what I was going to do with my own life. I believe that the engineering of life is the technology of this century, much as silicon was the technology of the last century. I think Eric framed quite nicely the history, but I want to tell you another little small history about the synthesis of life. About 14 years ago or so now, I got together with actually mostly a group of males from MIT who were computer scientists and bioengineers, and I was the token molecular biologist, also the token woman. And they framed the question, why is life so difficult to engineer? Why can't it be more like engineering, for example, a computer chip? And this led to the question of how can we make the engineering of life faster, more predictable, and cheaper. And we called this field, emerging field, synthetic biology, because it parallels, in a way, synthetic organic chemistry, which came 100 years ago and gave you the revolution of wearing things like nylon and pantyhose, et cetera. So what do we promise? Of course, we promise things like we're going to make lots of food, we're going to make plants that harvest sunlight better, we're going to make therapeutics for the third world or the developing world. And I think many of these things are possible. Why are they possible? What makes now different than, say, 30 years ago, where biotechnology was born right here with, for example, the production of insulin, human insulin in cells by Genetics Institute and Biogen. Now we have the ability to make DNA, which, as Eric said, is the substrate of life. And we can make it relatively cheaply, and we are going to make it ever and ever more cheaply very fast. And your government is supporting this effort in part, as is private industry. So we have the substrate of life. Our vision is that a student will be able to sit at a computer and be able to design a cell that does a specific function, order the DNA for that, have it come, and then show that the cell does something interesting. My original dream was to build cell-based computers. I realized they will never run as fast as your Apple computer, but they would do different things. The other thing we have at our fingertips is the internet. We have sequences of many, many organisms ranging from bacteria to humans to other mammals. This acts as substrate for how we will go about designing cells that, for example, can act as chemists or act as computers and make things that are interesting. So, from my own lab, what have we done that's interesting? So we've made what are called toy systems, where we prove that we can make cells that can remember past events. And actually, I never thought this would have any practicality until I gave a talk at a meeting, and then I got a phone call from NASA. And they said, we're interested in your cells that can remember whether they have been exposed to radiation or not. And I said, well, I thought NASA had no funding to do anything. And they said, oh, no, we have two programs. This was a couple of years ago. I think they have no programs now. But we have two programs. One is unmanned space flight, and the other is exposure to gamma radiation. And I have never in my life had this happen where the NASA representative said, we are just going to give you money to work on this. I didn't even have to write a grant. They just gave me money. That said, when it came time to write the grant, I didn't get the grant because they canceled the program. By the way, this should give you a flavor of the ups and downs of how us scientists have to work with regard to government funding, and we could come back to that. So lastly, let me finish with some examples of what we've done and what we'd like to do. So most recently, for example, we've made a mouse, no human experiments, a mouse gut bacteria that can tell us whether that mouse was ever exposed to a certain drug. Our dream going forward is that the mouse would, or the bacteria would respond to, say, inflammation and then make an anti-inflammatory. Now, why is this interesting? I don't know what the frequency of inflammatory bowel disease and Crohn's disease is, but I think it's pretty high, and it's a chronic disease. And chronic diseases are things that people suffer through their whole lives. So we think that by, for example, engineering your gut, that this may be a different approach to dealing with some of those problems. Going forward, I think that making plants that can harvest sunlight more efficiently is one of the biggest scientific problems and practical problems facing the Earth. If we can simply make the ability of plants to fix carbon 50% more efficient, we can help solve the global food problem. And then lastly, my favorite effort, Eric said, are you actually making life? We're trying. We're trying to make a programmable human artificial chromosome. We are trying to build it, as we say, from the ground up. And this challenges whether we know enough information to actually do that. Why do it? Well, it's interesting, it's a challenge. But also imagine if you could program your red cells. So red cells in their progenitor state have a nucleus, and then they lose it. And they live in your bloodstream for a relatively short period of time. So imagine if you could use your blood cells as a therapeutic, for example, to fight malaria. Can we program the red cells to do things that would be beneficial? And with that, I will stop and thank you for listening.
Fantastic. So our last panelist here is Glenn Cohen, who is professor of law at the Harvard Law School and co-director of the law school's Petrie Flom Center for Health, Law, Policy, Biotechnology, and Bioethics. I will say it's noted here that I came to know Glenn because I ended up filing an amicus brief with the Supreme Court in this breast cancer gene case as to whether or not human genes were patentable. And Glenn was my counsel, together with one other person, in filing this brief with the Supreme Court. And as an aside, we did win 9-0. Glenn.
What a batting average. Well, first, I was very lucky enough to be a fellow last year at the Radcliffe Institute working on something quite different, medical travel, medical tourism. I've got a book coming out in October, but a huge thanks to Liz and to Judy and to all of you who make the fellowship program possible. It was really an amazing year. Today I want to do 3 acts. Act 1, theory. Act 2, commerce. Act 3, politics. So first, Act 1, theory. When does life begin? I'm not going to answer that question, right? Because in part, I think it's the wrong question in the areas I'm going to talk about. In reproductive technology and abortion discourse, this question is frequently asked, as is the question, is X, a human being, an embryo, a human being, a fetus or an embryo, are they a human being? So is X alive? Is X a human being? I want to say those are the wrong questions. The right question is X a person? Living human being, those are scientific concepts. Person is a moral and a legal concept. You could have a view that all living human beings are persons. You could have a view that all only living human beings are persons. You could have a view that what it means to be a person, and a person is a bundle of certain rights, most importantly the right not to be killed willy-nilly, a right of inviolability. You could have a view, what's called a capacity X view, that there is a certain capacity or a bundle of capacities, rationality, emotion, ability to feel pain, that defines who gets rights from a moral and legal point of view. And under that definition, some animals might get rights and some humans might not get rights. But I want to suggest to you that that is the question we should be debating, not questions about living and questions about human being. With all due respect to my science colleagues, those questions are easy, but the question about who gets rights, much more difficult. Okay, theory. Act 2, commerce. So this is from something I wrote for the New England Journal with Elliot Dashi last year. There is a clinic in Los Angeles that kind of crossed a line that many people had not thought would be crossed in America anytime soon. And the line it crossed is it created batches of embryos made to order for sale. So essentially it found an egg donor and a sperm donor, put it together, generated 8 embryos or 9 or 10 at once, and then they sold the embryos one by one to potential buyers. So you would buy an embryo, and your embryo would have a genetic full sibling out there. And the question we were posed in this New England Journal of Medicine article is this ethical? The law doesn't prevent it. The laws governing the sale of body parts explicitly exempt sperm and egg. There is lively markets, especially here in Cambridge, for both of those things. Open up the Crimson and you'll see all the advertising. But the challenge we put to ourselves is to say, in a world where you can sell sperm, in a world you can sell eggs, and by the way, I know a lot about these markets and about how this goes about. I'm a real hoot at cocktail parties, right? But I want to say that in a world where you can sell those things, what is so different about being able to sell an embryo? Why should one be treated okay and one be crossing a line? Well, maybe you think one reason is you're worried about exploitation about the people who sell their eggs, for example. But that's a problem whether you're selling eggs for individual use or selling eggs to form an embryo. Maybe what you're concerned about is the feelings of the children that are born after this. This looks a little bit too much like selling children. Well, we say it's not like selling children, right? When you sell a child, who you sell it to will have an effect on its welfare. When you sell a gamete, an egg or a sperm, or sell an embryo, basically that will determine whether the embryo or the egg is produced for reproductive purposes and whether the child comes into existence or not. So it seems quite different. Maybe you think the problem is this corrupts the value of human nature, that we feel that human beings are being corrupted in this process. But then again, you might say, well, we allow the sale of the sperm, we allow the sale of eggs. You can buy both and put them together yourself. What is so different about putting them together at first? Maybe your concern is eugenics, that you think that this is about eugenics and buying designer babies. But the point we make is that actually the current markets where you can choose your egg donor and your sperm donor and put them together yourself are much more eugenic and much more bespoke. So at the end of the day, we came to the conclusion that if you think there's no problem with selling eggs and no problem with selling sperm, this extra step, although it feels icky and although a lot of people have problems with it, we're not quite sure what the problems are. If there is a problem, it has to do with the fact that the law hasn't caught up. We have embryo donation, quote unquote, embryo adoption as it's sometimes called in religious circles, and that is pretty well established. And what happens if a bank goes bankrupt, for example? What happens if someone wants to trace back their genetic parents? The law and the practice there is pretty well defined. The law and the practice for this new entity, this new venture, much less well defined. And we think if anything differentiates the cases, it's that. But happy to talk more in the Q&A. Okay, Act 3, politics. So I got a call in Mississippi a couple of years ago to show up at the University of Mississippi to debate the Personhood Initiative. So as you know, across America, there are a number of attempts to pass amendments suggesting that a human being from the time of conception to the time of fertilization is a legal person. They tried it in Colorado, it failed. They tried it in Alaska, it failed. In Mississippi, it was polling about 70 to 30 in favor of passage, so people started to get nervous. Well, it so happened my travel schedule didn't permit me to go to Mississippi, so I Skyped in, which is just as well because I didn't have to see the baby killer signs as I was kind of walking into the audience as my other colleagues who were there did. But afterwards, Jonathan Will and I wrote an op-ed in the New York Times, which got picked up and helped, I think to some extent, helped extinguish this vote where they ultimately lost. And what we said was, first of all, listen, if you're against abortion? Okay, you're against abortion. But understand this is not a great way to challenge abortion. It's not a great way to get a case before the Supreme Court. Because when you say from the moment of conception to the moment of fertilization, that is an incredibly ambiguous idea, as the scientists will tell you. Both is a legal matter. It's a process. There's not one moment where we say conception occurred. And depending on how you read those words, it may be the case that it got everything from certain forms of birth control to embryo discard as part of IVF to abortion, right? If you want to challenge abortion, pass a statute that criminalizes abortion, let the Supreme Court swat it down and do it that way, or fetal pain bills, which you've all written about. The other thing we said was, listen, it seems as though your constitution in Mississippi, which is what they were trying to amend, is self-executing. What that means is that every time in your statute it says the word person, all of a sudden fetuses and embryos are going to be persons too. So the doctor who performs your IVF cycle, or the bank that accidentally something goes wrong, has a power failure, and the embryos are destroyed, right? Those people may be liable for murder because they have either negligently or intentionally killed a person under Mississippi law. So what we said was this is a bad amendment because it's ambiguous, and this is a bad amendment because it does automatically what should be given a quite amount of thought. In Mississippi, we succeeded, but each of these goes on the ballot basically once a year. It's usually used as a get-out-the-vote mechanism. But I think that what's very interesting is one of these days, one of these things are going to pass, and I've got some thoughts on the constitutionality about it. When it comes to abortion, as long as the Supreme Court stays in its current configuration, which you never know, it's pretty clear that you could not prohibit abortion sui generis. You can kill it by 1,000 cuts, as they're trying to do with Texas admitting privileges and the like, but at least a strong prohibition on abortion like these personhood amendments would be no good. What about a constitutional right to reproduce, though, if you are infertile? Could your state prohibit IVF entirely, or could we adopt a solution that Germany has done, which says you can't freeze embryos? All the embryos you get, you either have to use for yourself or donate to another individual. Much, much less clear. Could the state ban certain kinds of birth control and leave other kinds of birth control available? Also much, much less clear. So I think what we're seeing is a very, and again, I'm a lawyer by training, which means I'm slightly amoral, even though I'm an ethicist, so I'm moral on that side. I'm kind of divided in myself. I'm like the Mini-Wheats commercial, right? But essentially what I want to say is my colleagues on the opposite side of the aisle, they're engaged in very good lawyering. I can admire aesthetically what they're up to because they've got a plan, they've got a strategy, and they are zeroing in on the abortion right. And one of the best allies people who want to maintain abortion as a right in America have are actually people who are infertile. And this was a mistaken, I think, gambit on their part because the opposition united those who are in favor of fertility treatments and those who are in favor of abortion. I have much more to say on that, but let me stop there.
Wow. Well, the Radcliffe Institute did a great job of pulling together quite a remarkable group of people here. We have about 25, 27 minutes for some discussion. We'll use some of it amongst the panel here, but you all should have at your places cards and should now be writing questions, ideally not essays, but questions, because we have a limited amount of time, and people will be coming through and collecting them. But I'm going to start off by just asking the panel about some hypotheticals here. Some I think are going to be easy, but even the fact that I think they're easy says something about where we all are. They may not be easy to you. David referred to this cool new technology that lets you edit genes, lets you change a couple letters in a gene. So I'm hoping this isn't the place we'll get into disagreement, but we'll see. There's a form of blindness that's due to a dominantly acting gene, like the dominant retinitis pigmentosum. You could imagine injecting a virus into the space of the eye that would go into many of the cells in the retina and cut out that bad actor, that dominant negative gene, and perhaps prevent retinitis pigmentosum by rewriting the genome of the cells in the eye.
You should all be filing a patent on this, by the way.
Oh, yeah, there are at least multiple companies in Cambridge that are attempting to do this. Quickie question, anybody find an ethical problem with that?
I would say no, because it's not going to be passed on to your progeny.
Exactly.
It's the same as taking any drug.
It's a drug. You take it yourself. You affect your own body. Glenn, the lawyer, of course, might find something.
So I don't find it unethical, but let me offer the argument of somebody who would find it unethical or offer an ethical objection.
Good.
And that is that there are many people out there who will either choose to remain blind or people who will not be able to afford this treatment. And when you diminish their political community of people who force our society to adapt to them, clickers when you cross the road, people to help them, right? They would say, you are diminishing my polity and my people, and I am losing the choice to be this way. Now again, I'm not particularly moved by the objection, but it's an objection.
And it is an objection, and so noted. But now let's make it, it's a serious objection. Access is an issue all the time. Questioning the validity of a community is, but it's not special per se to this technology. So now let me take it up a notch. So Pam makes these circuits, not just like old style, old fashioned, add one gene back in, that's so 1990s or something. Pam, no, I'm serious, that's old fashioned stuff. You're gonna make a circuit. Pam is gonna make a circuit that has 15 genes in it, and it's going to detect when the cell that is carrying that circuit, that set of 15 genes, has become cancerous. It's going to detect certain pathways have been turned on that say, I've become a cancer cell. And when it detects that, it's going to turn on another pathway that says commit suicide. That is, the cell commits suicide, not the individual. So I'm confident that Pam will be able to do that. Now, injecting that into some number of your cells isn't really the way to do it. The way to do it is to make sure that it's in all your cells. Well, too late for you, sorry. But we can do that for your kids. Pam will be offering sometime around 2024 this circuit of 15 genes that could be installed, and let's say installed safely without screwing up other genes, into the cell, which she claims will be able to put a little suicide switch in any cell that becomes cancerous. It will, of course, be passed down to your kids, and of course to their kids, and so on. Any ethical issues here?
I'll let you take that one, Linda.
I'm just going to ask how you do the clinical trial to show that there's no... How do you do the clinical trial for that, Pam? Because then we could use it for endometriosis also. Yeah. And I invite you to join my
Of course, we
center.
will diverge if we start talking about clinical trials, which is a big issue with using the engineering of biology. How are we going to do the clinical trials?
Probably in some other country.
No, I actually think Linda has the answer to this. That there's also another technology that she and I are involved in, which is making artificial organs, tissues, and transforming how we do clinical trials, and taking animals out of the equation, and maybe hopefully taking humans out.
So I'll pick up on that and agree with Pam that we are on a path of convergence. I think 2024 is maybe a little soon. You always put 10 years from now, but the idea...
I'm happy to stretch it out if it helps you with the ethical...
The idea is that animals don't represent people in so many ways that help us really predict what will happen in clinical trials. And I get a lot of money from the government, from drug companies, and everybody else to try to build more and more complicated, including the immune system, including ability to be dosed chronically with chemicals and things like that. So I can imagine that Pam's method could be in the future, this is stretching my own imagination, because you've got to have the stem cells, you've got to have all the lineages in the body, because a lot of times cancers may be influencing the early progenitor cells, and that's where it starts. We really don't know for sure. So I think if our methods ultimately approach real human organ mimics, then that Pam might be right. And
okay, so you're raising one ethical barrier, which is, and let me put it this way, that we probably won't be certain this is going to work based on any animal model or prior thing, and putting it into some human kids will constitute the ultimate proof that you got it right. You may think your models are good, you have a lot of good arguments, but we're not going to know till we put it in a person. So is that ethical to do that, knowing that we couldn't otherwise know?
I'm going to address that through my theragnostic bacteria that I call them, which is the idea of treating. Let's step away from cancer for a moment and step away from engineering your germline, and think about chronic diseases that people have to live with all the time, and when will the use of recombinant or engineered organisms become, no pun intended, palatable? Yeah. And I think it really has to do with the nature of the disease. Of course, cancer is an extreme case and has special laws, by the way, around the clinical trials, of course. But if you think about things like diarrhea in the third world, even travelers' diarrhea, which costs our country a fortune whenever the military goes overseas, there's 4 days out, that these are things that are transient in your body, and I think that the clinical trials would not be impossible to do, and I think there are drug companies that agree with that. So that might be the starting place for the acceptability of using these kinds of therapeutics, and as they become more acceptable, then maybe we will get to the possibility of engineering the germline.
Okay, so you're saying it's okay to put engineered bacteria in your gut because you're pretty comfortable you'd be able to wipe them out if it was a bad idea.
Or
you'd have an antibiotic against them.
You could do it in contained circumstances also. You could do it in the doctor's office.
I think it's important to emphasize also that we experiment, the world biomedical community experiments on humans all the time. That's really the fundamental nature of a clinical trial. And the extent to which we are willing to subject patients to risks, of course, strongly depends on the indication we're trying to treat. So if it's cancer, and even to build on Eric's example, if there are two parents that are both at risk of passing the same cancer-associated allele, cancer-associated gene onto their progeny is confirmed, the embryo is confirmed to have both bad copies, will now have a 80% chance of developing life-threatening cancer before the age of 50, then the barrier that most people would say that needs to be crossed before experimentation is truly out of the question, is higher than if it were a disease where there's no graveness, it's not life-threatening, etc.
All right, so you guys are telling me that the ethical issues here have to do with this testing, and maybe we should try giving you engineered bacteria, and then maybe we'll try curing cancer in the germline later. A lot of this is around the safety and ethics of testing people. But let me push it a little further. Pam also is going to work out some circuitry that improves memory. So not just curing cancer now, we're going to actually goose up those neurons that are involved retaining memories. And probably, as this spins out into industry, lots of other great new nifty ideas will be worked out for improving the human. I mean, we're really smart. We're going to think of a lot of ways to improve the human.
Just to protect Pam's inbox, these are all notional. Just to clarify.
And for those who are thinking about doing a postdoc in Pam's lab, they're not that notional. So, in any case,
thanks for giving me all these ideas.
That she's writing it down. So the point is, let's leave aside the safety questions. Suppose actually it were safe, we could do a clinical trial, none of that was the issue. Should we be doing this? Are there ethical issues about, quote, improving the human species based on somebody's idea of what's, quote, better?
So one set of people would argue that there is a difference between treatment and enhancement, the eradication of disease versus improving us from the baseline of where we are, and maybe the baseline is a range. Imagine the tallest person or the best memory. I have to say that I'm not particularly moved by that distinction. I think it's a problematic distinction. If we looked at the history of human beings, right, or even just the modern history of human beings, we have been, our memory, our height, our longevity have been quite different. And normal is just a very dangerous kind of baseline. But someone more sympathetic to this, like my colleague across the yard, Michael Sandel, might say, at some point we move away from the Aristotelian arete of what it is to be a human being, right? To be a human being is to be a particular kind of thing, and it's different from being a plant, it's different from being a computer, it's different from being an angel. And when we try to change our nature, we move away from what it is, the essence. I'm not particularly sympathetic to that point of view, but I imagine people that are may find find memory, when we get to the core of what makes us human, what we think is distinctive about us, that's when we start getting nervous.
So to sort of build on that, I see a lot of pierced ears in this room. I know at least one woman here has colored hair, artificially colored hair. I don't know about breast implants. I don't know about breast implants, which don't necessarily make the woman. So where does that distinction lie? Because arguably these are things that, by some definitions, are viewed as enhancements already. They're not genetic enhancements, but they're where is that boundary?
Well, the boundary might be roughly,
memory.
well, but the boundary is sort of, you put it out into the human population, it's passed on from generation to generation. Imagine the product recall. You decide it wasn't a good idea. How does your dealer tell you we'd like to take that back out? Anyway, Pam, did you?
Well, let me offer, I mean, these are trivial examples where we have altered the human race. For example, there were certain cultures where people were thought to be short, and then it just turned out that there was a vitamin D deficiency. They're not short anymore. And this also is important in agriculture as well, where we have been mucking around with agriculture for 5,000 years or more. And what's normal in a plant? Corn used to look a lot different.
Yeah.
And so we're constantly changing nature. I don't know if it's always for the better, but we're trying to hopefully do it for most of us for the better.
Most of us.
And I will
But I have a question from the audience here for you.
Qualify, when we met as synthetic biologists, and we've met many times, we always frame it as engineering biology for the good of the world.
Great. So that's a perfect lead-in to an audience question I've got here, which is, it's only a matter of time until a hacker/terrorist/crazy will be able to, quote, print a pandemic-grade pathogen. The technologies you all were talking about DNA sequencing and DNA synthesis getting really cheap and screwing things together so that they work. We all only have good uses up here, but maybe there are folks out there who have less good uses. What do you think is going to happen with this? Pandemic-grade pathogen coming?
The technology, I serve on a group called the Jasons, which is a group of academics that advise the government on matters of defense. So we talk a lot about biological warfare agents, the potential of them to be synthesized. The technology has already existed for quite a number of years to do this. It currently requires, sort of graduate school level laboratories at reasonable industrialized countries. I think we're already at a stage where we are more or less at the mercy of each other to not do what we can to create potentially deadly pandemics.
Well, that's reassuring.
I think we've actually been at that stage for a number of years. There was a paper published quite a few years ago now on the total synthesis of poliovirus. Okay, so we can make poliovirus. We can make any organism that exists at any point in its life cycle entirely as DNA or RNA. If that is true, if that criteria describes your organism, we can actually synthesize it in the lab. Now, most scientists don't rigorously consider viruses to be alive, but certainly the synthesis of poliovirus, which was injected into a mouse, kills the mouse, gives the mouse polio, was a little bit of a wake-up call to people that the ability to synthesize viruses, including ones that have dramatic implications for human health, is already upon us. Scientists have went to Brevig Mission, Alaska, okay, a place in Alaska in which almost everybody was wiped out by the Spanish flu of 1918 that killed hundreds of millions of people, the biggest human health disaster in history, dug up a Spanish flu victim from the permafrost, isolated viruses, sequenced the virus, and inserted genes that distinguish the Spanish flu from garden variety flu, and was able to resurrect the incredible virulence of the Spanish flu. That was also done a number of years ago. So we're already at an era where with fairly standard biotechnology can be used to resurrect pathogens, including ones that are responsible for the great loss of human health, and in principle propagate them. So that's why I say we're already sort of at the mercy.
So the questioner's already out of date here. Okay. Glenn?
I was just going to say, from an artistic point of view, so every age has its anxiety, right? It's always fascinating to see what that anxiety produces. And I think the current wave of zombie movies, World War Z, and also The Walking Dead and all of this, really is a reflection of this kind of a threat, the same way that we see a whole generation of art from the Cold War nuclear threat. It's very, very interesting.
I see. And as a lawyer, does the Second Amendment protect one's right to bear biological weapons?
Depends on who you ask, I would say.
Yes, it does, doesn't it? Okay,
what did
onward.
John Marshall think about biological weapons? Weigh in, right?
Exactly. That'll be a law school question, I'm sure, next semester. One of the questioners asks, how should we, that the handout for this panel says, how should we assess ... Whom do you include in we?
So I really empathize with that question because in this synthetic biology community, we've gone to great efforts to have a public discourse and engage the public. At the same time, there are many people that feel that the public is not being engaged and that the government is keeping things secret from us. And so we call this open access. We have something called open wetware. And so there's, much like the computer industry, there's a group that really believes in this open component to the engineering of biology. So I would encourage you to follow us.
So I'll weigh in from a little bit different perspective on a we, from the infertility angle based on some comments made here. It's a similar thing. There's a lot of debate about abortion, infertility rights. There's insurance mandates in Massachusetts to cover infertility. And I would argue that getting the public engaged helps flesh out what is society comfortable with. In particular, if you look at the amount of money spent on treating infertility patients relative to the amount of money trying to help women have healthier reproductive lives, it's almost a scandal in this country. Mm-hmm. And so these kind of debates make you stop and pause about what are we really doing. So we have engaging the we, as Pam suggests, by sometimes the extreme measures. One never likes to see people holding up signs, screaming outside of an abortion clinic, but it does slow down the race of scientists all excited about the discoveries to think about the consequences for humanity and how is it that as a society people really want to assimilate these things. You improve your memory, people are still going to probably go out and drink too much and do drugs and wreck all of that fabulous money they spent on enhancing their memory. How do
They'll be able to do more of it.
Yeah, they can still fund, you know.
We'll make a cell so you can take more drugs.
But I think the question, and this really was revealing, you go to the American Society of Reproductive Medicine, and there's so much money to be made, partly because of insurance mandates and so on treating infertility patients, that we kind of lose sight on how do we really think about the young girl, 15 years old, loves school, loves math, can't go to school and do math because her gynecologist is calling her crazy. So I think some of these debates, and that's, I gave a little bit more of a personal vignette, partly because it related to my Radcliffe experience, because there are ethical questions that get raised around this question you just brought up about creating viruses and so on that bring us back to the issues of reproduction that inform, I think, getting the public engaged and being okay as scientists for the people who just who feel it's icky. You got to listen to the people who feel it's icky because it's their world too.
I think also our experience with the stem cell story tells us the we is no longer the United States, and that whatever you announce as your policy decision, you're playing in a global game, right? And money, science, policy will flow towards, in many ways, less restrictive regimes, which is a very hard problem.
That's a very important point. China currently has, by many important metrics, greater biotechnology, genome sequencing, maybe even DNA biotechnology capability than the United States. So it is very important to point out that less restrictive countries don't really care what we think.
But I'd just like to mention, and Eric will know the formal name of this, that many of the international scientific associations have written a document about this very fact that you can download on the internet. I've forgotten the name of it, but and I'm sure Eric knows it. but it was written about 18 months ago, and there's a newer version as well. And you can all read that, and you will, and the Chinese Academy of Sciences is one of the signers of that.
Yeah, this is dual-use research of concern, yeah. So a question that plays off that from a questioner, and that maybe, this may be our last question, is, well, okay, who are we to assess? Let me take genetically modified foods. I bet most of us aren't so bothered by genetic modification of plants. In fact, we probably see it as a way to improve plants, improve nutrition, improve productivity. But there's a movement right now to have labeling laws. The FDA doesn't require labeling of GM foods because it says there's no difference between GM foods and non-GM foods. We're not going to require labeling, but some states want to pass laws that, in fact, by voter referendum, that will require labeling of GM foods. You for it or you're against it?
First of all, there's a question of whether it'll be legal or not, which has to do with what's called preemption, whether the federal food labeling
we could get out of it on, on federal preemption, all right,
Past
but
that's
too easy.
So to me, I would say, here's what. We have a very different attitude. You go to Europe, they are very serious about the GMO food. Our populace, not so serious. I guarantee you that if you pass a law that requires this labeling, people in America get more serious about it because they take it as a signal that says you should be paying attention to this just like you're paying attention to the calories, well, maybe not in New York, but elsewhere in the world where we've tried to do this kind of stuff. And my sense is if you really believe there's nothing to the distinction, passing a law in favor of this labeling tells people to pay attention to something they shouldn't be paying attention to.
And the scientists are going to try to remain silent on this question. It looks
We're running out the clock.
I actually, well, looking at the clock, but and perhaps you can, the difference between genetically modified, the definition of genetic modification is different in Europe than the US also, which is, so for example, and correct me if I'm wrong, my bacteria for the gut, because they don't contain, they only contain bacterial DNA, I think they are not considered genetically modified in the US.
Oh, cool.
I think that, whereas in Europe, the laws are very different. And also the ideology in Europe was, I don't want to belabor this, but was driven by issues around patents.
Oh yeah, no, it's the Monsanto
so it was economic.
history and
So what a lot of people don't know is that the attitude about GM in Europe it was an economic driver originally.
Indeed. Well, it seems to me that we could have continued for another hour or two on this subject. I don't know about you, but probably compared to, for those of you who are Radcliffe alumni, a lot has happened since you were last here. And it's moving at a terrifyingly fast pace. Now, I don't say terrifying to scare you, because almost all of the uses that we work on day to day are about dealing with serious diseases or other issues like energy, like food. There are incredibly important uses of all of this, but you can't work in this field without worrying about ethical issues of access, ethical issues about whether we are focusing on the right problems for people who matter or, for example, commercializing problems rather than focusing on what people really should be focusing on. There's just a huge range of things. It's exhilarating to work in this field, and as a parent or a person, one is also always aware of the sense of responsibility that comes in working in this field. I wish there were simple answers. There are not. But I'm so glad to have so many people who I presume are not mostly scientists care enough to come and participate in a discussion about this. And I want to thank the Radcliffe Institute for inviting this amazing panel to have this discussion.
Thank you.
Well, I don't think obviously I don't have to ask you to join me in thanking this fabulous panel for a really fascinating conversation. I feel like you led us kind of behind the scenes in the laboratory and in your private conversations. And that's really been very special. Thank you to Eric, thank you to Linda, to David, to Pam, and to Glenn for doing this. You are also made it very accessible to the audience, and for that, we are all very